Hello, in this video we will understand about the acetylation. We will also look at the deacetylation of the DNA and protein. So in this way the DNA acetylation controls gene regulation, for example the inhibition and the production of the protein, certain type of the protein. For example, the activation of the gene will lead to increase the inhibitor protein for inhibit the other gene activation. So is and regulate the gene for prevention of cancer even. So in this way, here is a person which that contain a cell. Each cell contain a same DNA in the form of chromosome. Chromosome contain histone protein and the DNA. DNA methylation will lead to silence the gene, while the demethyltransferase enzyme will lead to demethylation, and in this way the acetylation will lead to acyltransferase enzyme lead to oncogene suppressing gene will activate will lead to oncogene suppression, because this is the enzyme and the protein which are responsible for the inhibition of the other protein or the DNA activation. Remember about that thing, the acetyl group, basically which that's attached with a certain type of the protein also. And in this way, important things you should need to understand, the acetyl group will lead to activate the DNA, and methyl group will deactivate and silent. So in this way, the basically the overall the regulation of the methylation and acetylation will lead to prevention of the cancer. How? Because the acetylation activate certain type of the gene which that inhibit the oncogenes. So the epigenetic changes to the chromatin may result from development, environmental and drug and aging and diet, which that basically will change the acetylation, and on the basis of the requirement that certain type of the gene will activate due to the acyltransferase enzyme and the demethylation. The DNA basically here is the inaccessible gene and it is inactive. For example, the gene inactive, the DNA inaccessible gene are present in the form of histone protein wrapping around the histone protein, this DNA and this chromatin material. And basically which that basically the histone tail are also present which that attach if the acetyl group will lead to basically spin this histone protein will lead to wrapping of the DNA will lead to activate and produce the messenger RNA for the synthesis of the protein. This 4 unit, basically 4 type of the histone protein, H2B, histone 4, 2A and 3, collectively called a nucleosome. This nucleosome contain basically histone tail, and this tail basically active lysine residue. So if the acetylation will occur, so the DNA accessible gene will activate, and in this way the DNA will able to transcribe to translate the certain type of the protein. So the acetyl group is important for the production of the protein, while the methyl group will inhibit the gene transcription to produce the certain type of the protein. The DNA methylation and the chemicals modification of histone tail alter the spacing of nucleosome, and in this way the spacing will increase will lead to changes the gene expression. In this way, where the space are present, the DNA accessible gene activation. In this way, the certain type of the gene will transcribe to mRNA to produce a messenger RNA to produce protein by a translation. So our required, if for example you are working hard in the gym, so the synthesis of the protein is crucial. So that is why you will synthesize more and more actin and myosin protein to build the muscle. Protein deacetylation by SIRT1 gene, which that is important for anti-aging process. Remember, the NAD plus is involving for as a basically is the this is molecule act as electron acceptor during the deacetylation, and in this here is the lysine residue which that's contain acetyl group. So it will donate, and the NAD plus will accept this proton in the form of proton, and in this way acetyl group will accept in the presence of water. So the SIRT1 gene. SIRT1 gene is a family of enzyme that use NAD plus as a cofactor to remove acetyl group from lysine. So from the lysine is a protein which that basically deacetylation will lead to free the lysine residue containing deacetylated protein, and in this way the 2-oxyacetyl ADP will be separate with the AMP, adenosine monophosphate, attached with the basically here you can see the anything. So in this way the NAD plus will break down, will produce the NAM, nicotinamide adenine mononucleotide, while the nicotinamide adenine dinucleotide is basically the 2. So in this way, remember about that things, this formation and accept the acetyl group in the glucose carbon number 2, and that is why it's known as a 2-oxyacetyl ADP. So the SIRT1 is a family of enzymes that use NAD plus as a cofactor to remove acetyl group from lysine. While the NAD plus, this is molecule, act as electron acceptor during the deacetylation. But the 2-oxyacetyl ADP ribose by product of deacetylation reaction, and in this way this is the product, but in this way the protein will be separate, which the deacetylated, no acetyl group will be added. In this way this protein can be inactive, for example the inactive enzyme. So enzyme will not work properly, but the again acetylation will lead to trigger the function of the enzyme. Let's understand about the protein acetylation. In this is the polypeptide which that contain and carbon alpha which that attach with the lysine residue. So the lysine residue is responsible for accepting the acetyl-CoA and basically acetyl group. So acetylation will occur. In this is the normal alpha acetyl transferase enzyme used to the acetyl-CoA to convert CoA, and in this way donate the acetyl group in the end of the alpha carbon. So the lysine residue is basically contain alkyl group is here, and in this the NH will attach with the CoA, acetyl group COCH3. In this way, basically remember about that things. The H proton will be accepted by the enzyme, and in this way remember this is the lysine. Lysine basically is here you can see, and in this way lysine will attach with the nitrogen side, the acetyl group attach, and in this way acetyl-CoA will use, and the cats will be used as an enzyme. And on the other hand the deacetylation also occur, will be remove this acetyl group to convert the lysine, deacetylated lysine, and here is the acetylation. Acetylated lysine which that contain a acetyl group. So I hope you make sense about that. This is the protein deacetylation and the protein acetylation. So protein acetylation will lead to activate the certain type of the enzyme, the globular protein, and other type of the protein which that active, and the folding of the protein and the activation. On the other hand, the acetyl-CoA will basically also donate to the Krebs cycle to produce NADH, FADH2. And this basically is the acetylation of the in the Krebs cycle the compound. So the epigenetic changes may result in basically cancer, diabetes, mental disorder, a lot more.